Hi everyone! In this video I'm going to be going through the step-by-step process that is involved in hearing. So we're going to be looking at the ear, specifically this region right here, including the inner ear, the middle, and the outer ear. And we're going to be zooming in also on this structure right here, the cochlea. We're going to be taking the cochlea and imagining that it's stretching out so that it's more like a straight line because it's all coiled up normally like a snail shell. We're going to take it and we're going to just kind of unwind it a little bit. Okay, so what we're going to be drawing is this. So we have the outer ear, which includes our auditory canal and our tympanic membrane. Okay, the middle ear is going to have our ossicles, which I'll get to, and our eustachian tube. And then of course our inner ear, I'm going to just ignore the other structures in the inner ear because they are not involved in hearing, and this video is all about hearing. So I've taken the cochlea, which normally is kind of coiled up like a snail shell, and I've cut it in half and taken off the front half so we can see inside of it. So that is what the cochlea would look like, all kind of unwound and cut in half. So of course we have the ossicles. Remember, for Bio 30, you just have to remember that they're called the ossicles. You don't need to remember the three individual names. The ossicles connect to this circle, which is called the oval window. Okay, it's kind of the first part of the cochlea. And then we also have the round window on the bottom side of the cochlea. And then this structure that kind of separates the top and the bottom half of the cochlea is called the basilar membrane. So we're going to be looking at this structure really specifically because that is where hearing takes place. So I'm going to be going through and drawing kind of this process, and I'll number them, and then at the end of the video I'm going to be talking about what each number represents. So while I'm talking about it, I will be mentioning the things that you're going to eventually write down, but on the last slide I will actually go through the step-by-step process. So make sure you leave room for not only the diagram but also the step-by-step process. So number one is the vibrations of sound, the sound waves coming in through the auditory canal and hitting the tympanic membrane, causing it to vibrate like a drum. Okay, once that happens, because the ossicles are connected to the tympanic membrane or the eardrum, and if it starts to vibrate, it's going to start to move the ossicles, and the ossicles are in charge of amplifying those vibrations, which means that it increases them in magnitude. So that's number two, going through the ossicles, okay, just amplifying those vibrations. Eventually, they're going to strike the oval window. So this last ossicle will strike the oval window and cause little ripples to happen inside the cochlea. The cochlea is filled with fluid, so if we're having movements happen right here, it's going to cause the fluid inside the cochlea to start to ripple. Just like if I threw a stone into a pond, you would see ripples after that stone falls into the water. So that's the third thing that happens. Then we're going to see that ripple travel down the cochlea. Now it depends on the pitch of the sound that we're interpreting. If it's a really high pitched sound, it will hit here on the cochlea. If it's a low pitched sound, okay, it will travel farther and hit the bottom part of the cochlea. So depending on the pitch of our sound that we're looking at, okay, it depends on where on the basilar membrane it will actually hit. Okay, let's say that this is a fairly high pitched noise, hence why it's hitting fairly high up on the cochlea. So it's going to go through the liquid and eventually hit this basilar membrane and cause it to be pushed down. So when this happens, I'm going to actually zoom in on the basilar membrane. I'm going to imagine that I'm cutting this open and kind of peeling it back and looking at it on the inside. I'm going to zoom in on it. So inside the basilar membrane, it's actually called the organ of Corti. Now, I don't know about you, but that's the greatest name I've ever heard for a Pirates of the Caribbean movie or possibly a Harry Potter movie. So my favorite name in all of Bio 30 is this organ of Corti, and it's made up of two different membranes. So we have the top membrane, which would be seen right here, and then on the bottom membrane we have our stereocilia, which are basically specialized hair cells. If this gets pushed down, it's going to basically cause these little tiny hairs to bend. So when this little ripple happens where we push it down, okay, this is going to push down into the stereocilia, and it's going to cause the stereocilia to bend. When they bend, it's going to start an impulse down the nerve cell which connects to our brain. Okay, it's going to start an action potential which travels all the way to our brain, and we would interpret that sound depending on the pitch, okay, in our brain in a certain way. So after that happens, the last step that needs to happen is it needs to just go through the round window. The round window will just kind of allow this ripple to go out a little bit so that it allows a little bit of movement. Okay, if this structure had no sort of membrane that could move in and out, it wouldn't allow movement in the cochlea at all because it would be jam-packed with liquid and there wouldn't be any room for movement at all. Okay, so that's what the round window allows it to do. It just kind of pushes out so that the ripple can kind of just continue on, and then it would just be equalizing the pressure within that structure. This last step, number 6, does not have anything to do with hearing. Okay, once this basilar membrane goes and we send that stereocilia bending, we send that impulse to the brain, okay, that's the end of the hearing. This is just basically finishing off the ripple so it has somewhere to go. So I'm just going to show you guys a different diagram of the ear just because obviously my diagram isn't amazing. So this was the auditory canal, the tympanic membrane, our 3 ossicles hitting the oval window. Okay, this is the cochlea. Now this is a picture of inside the basilar membrane, okay, the structure called the organ of Corti. There's these 2 membranes, and inside the bottom one there are some stereocilia or hair cells. If this one pushes down, it's going to cause the hair cells to bend. Okay, and send an impulse to our brain. And then this was just showing you if I unstretched the cochlea, this is kind of what it would look like. Again, really close to the oval window and the round window. If it's a high-pitched noise, it would be going across the basilar membrane there. If it's a low-pitched noise, it travels all the way to the very end. And then just another diagram again, the cochlea cut in half. We see the top and the bottom chambers, and then inside the middle this would be the basilar membrane, and inside that is the organ of Corti. Okay, again, just zooming in on it, we have a top and a bottom part of that. We have stereocilia. If this pushes down, it causes the stereocilia to bend, and it's going to send an impulse to our brain. So let's just go through quickly the step-by-step process of hearing. So the very first thing is the sound wave is going to travel through our auditory canal and hit the tympanic membrane, causing it to vibrate. Okay, those vibrations will travel to the ossicles, and the ossicles will amplify them. Okay, the last ossicle will strike the oval window like a drum and cause the ripples to go through the fluid in the cochlea, because the oval window will be pushing into the fluid, causing ripples to happen. Now the ripples will travel through the cochlea, through that fluid, and hit somewhere on the basilar membrane. Remember, dependent on pitch. So if it's super low pitch, it'll go farther. If it's high pitch, it will not travel as far. Okay, once it hits the basilar membrane, it's going to push down that top membrane and cause the stereocilia found within the organ of Corti to bend, which starts an action potential on those nerve cells and eventually travels to the brain. And then the last part of this, remember, nothing to do with actual hearing. The ripple has to go somewhere, so it's going to hit the round window to just equalize that pressure. So that is the end of our steps of hearing. Okay, you should know how to draw the diagram, and you should know the step-by-step process. Okay, this is one of the key pieces to chapter 12. So if you don't understand this, you have to make sure that you come and ask me questions. Otherwise, you're going to be missing one of the key pieces to this chapter, and obviously missing not only on the drawing quiz, but also on the chapter 12 exam. This is a really important process that we need to know for the chapter 12 exam. So if you have any questions, please don't hesitate to ask. Okay, if not, I'll just see you in class tomorrow. Okay, we will have to draw this out for a drawing quiz, but I'll see you guys all tomorrow. Have a wonderful evening. Good night.